Hello, am I audible? Hello, am I audible? Okay, so I don't think there are any issues with the audio feed. Okay, we will start with our next lecture, that is the fifth part of the origin of life. Okay, before starting, let's see what were the points which we have already seen. Okay, see that we have seen these different theories of the origin. Okay, in that we have seen the operance theory. Okay, so it was the chemogeny, biogeny was there. Okay, we have seen an experimental model to prove the operance, that is the Miller's apparatus. Then we have seen the origin of this life, chemical origin of life. What we have seen, we have seen origin of different organic compounds. Okay, hydrocarbons, carbohydrates, different derivatives of those hydrocarbons, glycerol, fatty acid, lipids. Okay, we have summarized all that organic formation into this one diagram. Okay, then we have called about the complexation of those simpler compounds. Okay, जो simpler compounds है उनका complex form का हमने discuss किया, how it's going to be formed. Okay, then we have seen the two these what we can say options or two possibilities from which the first live cell has been formed. Okay, हमने क्या देखा, पहले तो थेरीज वगैरह देखा पूरा. ओके, उसके बाद हमने मिलर्स apparatus देखा, राइट. देन हमने ये देखा कि नाउ ऑर्गेनिक कंपाउंड्स दैट इज द कीमोजेनी. ओके, केमिकल सिंथेसाइज होंगे पूरे अक्रॉस द अर्थ. ओके, हाइड्रोकार्बन होंगे, अमीनो एसिड होंगे, प्रोटीन्स होंगे, फैट्स होंगे, राइट. सो ये सब केमिकल फॉर्म होने के बाद हमने क्या देखा कि इन केमिकल्स का, सिंपलर केमिकल्स का भी कॉम्प्लेक्स केमिकल्स बनेंगे. ठीक है. So once this complexation of chemicals, polymerization occur, condensation occurs, then we have different chemical compounds onto the earth. Okay, अभी earth में बहुत सारे chemical compounds है which can be utilized to form a first living cell. Okay, अभी हमारे पास derivation होने के बाद बहुत सारे cells हैं. Now these we can use for the formation of what? A living cell, a living cell. Okay. Now to form that living cell or to form that living body, there are two options we had: coacervate and microsphere. Okay. हमने already देखा कि coacervates also behave like biological system. The microsphere also behaves like a biological system. Out of them, okay, इन दोनों के बीच में, coacervate और microsphere के बीच में, माइक्रोस्फीयर के ज्यादा चांसेस है दैट माइक्रोस्फीयर हैज मोर कैरेक्टरिस्टिक विच एक्चुअली व्हाट फिट्स टू दिस क्राइटेरिया कि दोनों में से कौन ज्यादा पहले अगर सेल बना होगा तो माइक्रोस्फीयर ज्यादा बना होगा. ओके, इट इज एन फोर रनर. ओके, नाउ दिस टुडेज लेक्चर. ओके, आज के लेक्चर में हमें क्या लिखना है? ठीक है, दैट इज द थर्ड स्टेप. ओके, सी वंस वी हैव फॉर्म द डिफरेंट केमिकल कंपाउंड्स. ओके, एक बार हमने पूरे केमिकल कंपाउंड्स, ऑर्गेनिक कंपाउंड्स फॉर्म किए. ठीक है, नेक्स्ट फेज क्या होगा? To form a cell, to form a living cell, to form DNA और to form न्यूक्लिक एसिड. ओके, एक बार हमने केमिकल्स तो बना लिए पूरे केमिकल असेंबली के सेल के, पर अभी अगर पूरा केमिकल असेंबली सेल बनाना है तो ऑलरेडी हमें क्या लगेगा, जो सेंट्रल ब्रेन जो है सेल का, व्हाट इज द सेंट्रल ब्रेन ऑफ द सेल इज दैट वी रिक्वायर, which is what, which is this DNA. Okay, so now the next third phase is the formation of this nucleic acids or the naked genes. Okay, what will happen now? Now there will be synthesis of different nucleotides, DNA, RNA, from all the nucleotides which are going to be formed from what? From this already synthesized chemicals. Okay, जो chemogeny में जो chemicals बने हैं उससे अभी क्या होगा? What this it's going to be formation of these different chemicals, that is the nucleic acid and naked gene. Okay, see, with the hydrocarbons, with the carbohydrates, with the lipids, fats, proteins which we have already synthesized. Okay, ये जो पूरे कार्बोहाइड्रेट वगैरह जो हमने ऑलरेडी सिंथेसाइज किए हुए हैं, ओके, उससे हम क्या कर सकते हैं? राइट नाउ वी कैन फॉर्म अ काइंड ऑफ व्हाट, अ साइक लाइक स्ट्रक्चर्स. ओके, एक सेल मेम्ब्रेन जैसे कंपोनेंट्स तो फॉर्म कर सकते हैं राइट नाउ. ओके, with all of these chemicals. Okay, but what we require, we now want this nucleus and the DNA which is there in that nucleus. Okay, ये जो न्यूक्लियस है और उसके अंदर का जो डीएनए है वो हमें चाहिए. ओके, बट ऑब्वियसली इट कैन नॉट बी डायरेक्टली सिंथेसाइज्ड विद प्रोटीन्स. ओके, सीधा प्रोटीन से तो नहीं बनेगा ना. ओके, वी वांट द बिल्डिंग ब्लॉक्स ऑफ दिस डीएनए एंड आरएनए व्हिच आर व्हाट, दिस न्यूक्लिक एसिड. ओके, उसको ऑलरेडी क्या कहा जाता है, नेकेड जीन्स। ओके, सो दिस इज द नेक्स्ट स्टेप आफ्टर दिस। ओके, now abhi what is the case is that, okay, there are certain scientists, some scientists are there who believe, okay, वो क्या सोचते हैं कि पहले आरएनए बना, बहुत सारे टाइप्स ऑफ आरएनए बने। ओके, फर्स्ट डिफरेंट टाइप्स ऑफ आरएनएज आर फॉर्म, mRNA, tRNA, rRNA, all these kind of RNAs are formed. Then all these mRNA, tRNA, rRNA combined and they started to synthesize what, different proteins. Okay, ये दो theories है। Okay, so the scientist who believes that the RNAs are synthesized first is called as RNA world. The scientist who believes now the second type of scientists, are second branch of these this thinking is what, that first the proteins are formed, पहले तो सब proteins बने। Okay, then from these proteins have ultimately what led to the formation of these different RNA and DNA. Okay, see ultimately to for this different process of this transcription, translation, okay, हमें क्या चाहिए, okay, हमें we also require RNAs also, we also require proteins also, right? But some scientists said that पूरे RNA पहले बने, then protein synthesis started, okay, and some scientists are saying what, only the proteins are synthesized first, then the RNA or DNA formed later, right? So the first one is the what, RNA world hypothesis। इसमें क्या हो रहा है, the RNAs are synthesized first। ये जो hypothesis दिया था that the RNA is synthesized first, it is given by the Orgel, Crick and Woese। Okay, Orgel, Crick and Woese ने ये क्या दिया था, the hypothesis दिया था। ठीक है, next is what, RNA can show different functions। Okay, RNA क्या दिखा सकता है, okay, सोनू सिंह is saying yes। Okay, yes, good morning सोनू सिंह। ठीक है, so RNA can shows different functions. Okay, as see why it is said that it could, why some scientists believe that the RNA is formed first, because it is observed that the RNAs are itself capable of performing multiple functions. Okay, these functions are like that, they are catalysts for different reactions which are occurring in that cell. Okay, they can also regulate some of these genes or DNAs or the RNA, right? They can also form these different NAD, FAD, nicotinamide adenine phosphate, जो है ऐसे बहुत सारे जो components है which are ultimately required for different cellular cycles, they are also going to be formed from this। Okay, and these are also going to be very much important into the protein synthesis। Okay, see these are some of the points which actually what, emphasize the RNA's functions in different cellular activities। ठीक है, see ultimately as I have told you ना कि ये एक shorter version है जो मेरे पूरे detail batch का है, so that's why I have given some of the functions। Okay, actually it has some more functions to it। Okay, that is why I said कि अगर आपको detail वाले चाहिए तो आपको ये 15-20 मिनट में या फिर आधे घंटे में मैं कंप्लीट नहीं कर सकता। ओके, देयर आई गिव लॉट ऑफ टाइम, 1-2 घंटे लेता हूँ मैं उस पेड बैच में। ओके, सो दोज हु डोंट वांट, दे कैन जस्ट स्टिक विद दिस यूट्यूब लेक्चर प्लस नोट्स। ओके, तब भी सफाइस रहेगा। ठीक है, दोज हु कैन ज्वाइन, वो पेड बैच ज्वाइन करो, और पेड बैच अगर ज्वाइन किए तो यूट्यूब मत देखो, बिकॉज इट विल जस्ट बी द रिवीजन ऑफ द सेम सेशन। ठीक है, so these are the different functions of that RNA। So based on this RNA world hypothesis has been put forth that as the RNAs are itself able to function different function, तो पहले RNA बना। ठीक है, प्रोटीन फर्स्ट वाले that is theory given by the SW Fox। okay, वो क्या बना है, that the प्रोटीन कैटालिटिक जो systems है बहुत सारे which are required for the synthesis of this RNA, DNA or other components are synthesized first। okay, ये पहले बनी। okay, so these are the two hypotheses are there which are the series of this RNA and protein। ठीक है, now we will also have to see that ये जो प्रोटीन सिंथेसिस है वो कैसे इवॉल्व हुआ। ठीक है, द इवोल्यूशन इज क्वाइट सिंपल। ओके, जो अमीनो एसिड्स व्हिच वेर देयर, नाउ व्हाटएवर द अमीनो एसिड्स वेर देयर, ओके, फर्स्ट द पॉलीमराइजेशन ऑफ दोज अमीनो एसिड्स स्टार्ट। ओके, वो पॉलीमराइज हो गए। ओके, देन द डिफरेंट अमीनो एसिड्स विल अल्टीमेटली, इफ वी जस्ट पॉलीमराइज द अमीनो एसिड, इफ वी जस्ट कंबाइन द अमीनो एसिड, सो अल्टीमेटली हमें क्या मिलता है, पॉलीपेप्टाइड। okay, peptide bond ज्यादा होंगे उस entire amino this what we can say polymers में, right? So that's why it is called as polypeptide, right? So polypeptide form होंगे, right? Then once these polypeptides are formed, then these different types of RNAs, okay, mRNAs, rRNAs, tRNAs will form। okay, and once these mRNA, rRNA, tRNAs are going to be formed, they are going to carry out this entire protein synthesis function। okay, so this is how is going to be evolved। okay, this is going to be the sequence, amino acids से peptide बनेंगे, polypeptide बनेंगे, then mRNA, RNA form होंगे, और they will combinedly carry out what, protein synthesis, right? नेक्स्ट फेज इज इवोल्यूशन ऑफ डीएनए। ठीक है, इवोल्यूशन ऑफ डीएनए में क्या बोला जाता है, दैट फर्स्ट दी आरएनए वाज देयर। okay, single stranded RNA was there, okay, and that single stranded RNA later on might have came, okay, in contact with the other RNAs, ठीक है. Evolution of DNA में क्या बोला गया है कि पहले RNA था single stranded, वो सिंगल स्टैंडर्ड आरएनए किसी और सिंगल स्टैंडर्ड आरएनए के कांटेक्ट में आ गया, ओके, और मे बी द सिंगल स्टैंडर्ड आरएनए जो है, ओके, उसके कॉम्प्लीमेंट्री बेसिस सिंथेसाइज हुए उसी स्टैंड पे, ओके, एंड अल्टीमेटली दैट सिंगल स्टैंडर्ड this what the RNA converted into the double stranded RNA, and this it got coiled, and ultimately what we have this double stranded DNA, helical structure of the double stranded DNA, okay. This is how the evolution of DNA might have occurred, okay. नेक्स्ट is formation of the different nucleoproteins or the protoboans, okay. इसमें एग्जांपल क्या है, नेक्स्ट प्रोटोबायंस में एग्जांपल क्या है, अर्ली राइबोसोम्स जो है which are the proto-ribosomes, वो फॉर्म हुए फर्स्ट, ओके, देन दी प्रोटोवायरसेस which are what free living genes are formed, ओके. इसके बाद में ये जो वायरस हम जो बात कर रहे हैं वो इसके बाद फॉर्म हुआ, ओके, ओके, दी वायरल जीन्स और दी फ्री लिविंग जीन्स आर फॉर्म लेटर दिस, ओके. So now these were the different parts or different points which we'll have to explain in the chemogeny. Okay, chemicals में हमने amino acid form किए, we have formed these different nucleic acid, we have formed hydrocarbons, we have formed different carbohydrates, proteins, fats, DNA we have formed, nucleoproteins we have formed, right? Now the time to study this biogeny. Okay, अभी biogeny means what, life कैसे evolve हुई है, ठीक है. So formation of these organisms or the whatever this eobionts is there, ना ये organism कैसे form हुए, इसके लिए two theories है. There are two theories to explain that how these organisms are formed. Okay, first theory is what, Horowitz theory. Okay, पहला जो scientist है Horowitz, उसने concept दिया first Horowitz concept. Second is the Oparin concept. Okay, what is the difference in between two? Horowitz concept में क्या बोला गया है that first the nucleo, see, ultimately nucleoproteins are going to be important for formation of the first living cell because they are going to ultimately form the nucleus and DNA, right? So that's why both of them consider that the, if final cell or final primitive cell is having nucleoprotein inside them, then that is what, the proto cell, okay. That means what, this cell is only going to become what, living cell. Both of them, दोनों का मानना यही है, ठीक है. But the only the way in which this cell will form will different. Horowitz क्या बोला, what the Horowitz is talking about, he is saying that the first such nucleoproteins will come together, okay, then there will be formation of different sheath will be there around them, okay. Why the formation of sheath will be there, because the required component to form that sheath is already present in that hot soup, okay. जो सी है, हॉट सूप सी है, उसमें ऑलरेडी ये जो पूरे कंपोनेंट है, ओके, वो क्या करेंगे, they will form different sheets around these what, nucleoproteins, okay. Maybe one nucleoprotein will be enclosed, maybe two enclosed, three enclosed, okay, that will vary, right. So once that sheath is formed, so ultimately they will form a structure like this. वो फिर उनका उस sheath का क्या बनेगा, cell membrane या फिर cell wall बनेगा, ठीक है. Now once that cell wall is formed, that is, and it will have the nucleoproteins inside it, so that is what, a first primitive cell. अभी सिर्फ क्या है ना, इसमें सिर्फ जान डालना बाकी है, okay. Now what is required, just to what, to add a life to it, okay. That means what, these nucleoproteins will form a nucleus, then it will become what, a functional living first living cell, ठीक है. So ये बोला गया Horowitz ने. Oparin क्या बोलता है, Oparin is saying that first these different nutrient molecules will combine and will form a sheath like structures, okay. ऐसे vessel या फिर sac like structures form करेंगे, okay. Then what will happen, either these nucleoproteins will be synthesized in this, okay, or they will add it into it, okay, from external, ठीक है. Ultimately क्या form, ultimately the same structure is getting formed, right. So these are the two different aspects are there, okay. First Horowitz concept is saying that the nucleoproteins will combine and the sheath will grow around them, and the Oparin the concept is saying that the first the sheath and the sac-like or cell membrane or cell wall will form first, and then this nucleoprotein will develop into it, okay. So दो concepts है that how this eobionts or this first primary organism is formed, right. Next is the evolution of the modes of nutrition. अभी हमने क्या देखा, we have just formed the cell here, okay. But अभी ये cell evolve होने के लिए obviously they will require what, nutrition, okay. तो वो nutrition कैसे evolve हुआ है, okay, how the evolution of nutrition might have occurred, okay. See, first is the chemoautotrophs, means what, first these primitive cells developed a characteristic or develop an habit of what, Chemoheterotrophs means what? To feed on the chemicals. Okay. See, they feed on the chemicals. Okay. Suppose they have almost exhausted all the chemicals in that hot soup. Okay. Hot soup C जो है, okay, उसमें से जो पूरे C में जो chemical component है, which these first primitive cells can eat. Suppose they have eaten all of them. Okay. So now there is scarcity of food. Okay. So what will happen? Then it will start these different modes of nutrition. Which are these? These are the parasitism. They will, ये क्या करेंगे? What they will do? They will attach themselves with other living cells, okay, and they will feed on them only, right? Saprophytism क्या करेंगे? Now these will only feed on the dead microbes, okay, or dead living cell, okay, जो कुछ भी dead होंगे बाकी के उसमें से उसके ऊपर ये feed होंगे, ठीक है? Predation means what? They will just decide to directly eat live or directly attack and kill other living cells and eat them, okay. Chemoautotrophs means क्या होगा? Now they will just start to synthesize their own chemical food. Okay, वो खुद का food खुद बनाएंगे, ठीक है? So these are different modes of nutrition will develop. Okay, ये तो chemoautotroph हो गया. Okay, second is what? Photoautotrophs. Okay, they can make their own food. So there are two types of, okay, with the help of sunlight, okay, or with the help of radiation, or with the help of the other energy, okay, photovoltaic energy के साथ ये खुद का खाना बनाएंगे, right? So photoautotrophs are there, okay, but here there are two types are there. Anaerobic photoautotrophs are there and aerobic photoautotrophs are there, okay. So anaerobic क्या है? Okay, so these are first chlorophyll containing photoautotrophs are there, okay. वो oxygens का utilization नहीं कर सकते या फिर they are not utilizing or releasing oxygen, ठीक है. So anaerobic है ये, ठीक है. So here this chemo, see this is the diagram of the chemoautotrophs. ये जो हमने chemoautotrophs किया ना यहाँ पर, okay. इसके लिए ये reactions है. Glucose will be fermented into the ethyl alcohol, carbon dioxide and energy will be formed. Okay, this is how they will feed. Okay, ये जो anaerobic photoautotrophs है ना, this will be feed like this. Okay, they will utilize the carbon dioxide. They will not utilize water, but they will utilize the hydrogen sulfide. Okay, and then they will produce the glucose, water, and sulfur. Okay, so this is how they will adhere. Here there is no what interaction of oxygen generate नहीं कर रहा है यहाँ पर, ठीक है. But the aerobic photoautotrophs क्या, what they will do? They will now produce the oxygen. अभी ये क्या करेंगे? Oxygen produce करेंगे, ठीक है. Now once this aerobic photoautotrophs produce oxygen while their metabolism or while their nutrition, then what will happen? Then throughout the earth the oxygen will start to release into the atmosphere, ठीक है. So that now the oxygen will start to fill this atmosphere, ठीक है. See, because of that presence of, because of that increasing concentration of oxygen, what will happen? See, earlier we have seen that the earlier atmosphere was was reducing in nature, okay, because of the presence of hydrogen and similar gases, okay. Earlier जो atmosphere था वो क्या था? Reducing था, okay. Now when this oxygen will start to get added in this entire atmosphere, okay, it will convert from reducing to what? Oxidizing. अभी क्या होगा? Because of the increased concentration of oxygen and the reaction which will occur with this oxygen, reducing atmosphere will be converted into the oxidizing atmosphere, okay. Now that oxidizing atmosphere will again be conducive to for the what? Growth of the life on the earth, right? So now this is the part, okay, which we will study for today. Okay, आपको अब तक का ये जो पार्ट है वो यहीं पर हम स्टॉप करते, वी विल कंप्लीट दिस ओरिजिन लाइफ ऑफ लाइफ रिमेनिंग पॉइंट इन द नेक्स्ट लेक्चर, ठीक है. बट द नेक्स्ट लेक्चर विल बी ऑन मंडे डायरेक्टली, ओके. अभी कल सैटरडे संडे है तो यूट्यूब पे कोई सेशंस नहीं रहेंगे. नेक्स्ट सेशन विल बी देयर डायरेक्टली ऑन मंडे, ओके. मंडे 5 PM पर रहेगा, ठीक है. So I don't see queries are there, so we'll stop here. We will meet next Monday, coming Monday. Till then, have a great day. Bye bye.